Thank you very much. So we're going to spend the next hour and a half or so talking about how to use isotopes to study animal ecology. And Turi's lecture this morning was really nice lead-in because you got an appreciation for sort of what tissues you can measure and what that tells you about what animals did over the past days to weeks to months to years. And we call that isotopic incorporation. And it's a really obviously detailed and complex story, but it's really important to keep that in mind when you're doing your studies and designing your studies. So I'm going to give you a quick outline. I'm going to give you a brief introduction of kind of myself, a new project, how I'm spending my startup cash. Carbon and nitrogen isotope discrimination is the main thing that we're going to focus on today. So we're going to basically, I'm going to get through the physiology that's associated with the differences you see between animals and the things they consume. That's it, okay? And that's really all. That difference, that isotopic difference that we call discrimination, is mediated by physiology. It's mediated by stuff that's going on inside your bodies right now as you metabolize the stuff that you consumed today, this morning, and yesterday, and weeks ago to build and maintain your tissues, all right? And then if we have time, I'm going to do a Craig Cook sort of inspired idea, choose your own adventure, talk about some recent projects in my lab, one on gut microbiomes and the other ones on hydrogen isotope values and how you could use this as a tracer of resource use, not just water, drinking water, like we heard a lot about this morning, but also to trace food. And so you'll get to decide which one we want to do. So I did my PhD at UC Santa Cruz. I was actually in earth sciences. I'm in a biology department now, but I don't have a degree in biology, so don't tell my colleagues that. But I got my degree in earth sciences. I studied historic ecology, or historical ecology. It really is the past distribution, behavior, and ecological role of relict animal populations. What I mean by relict animal populations are populations that live in small populations today in comparison to what they were tens or hundreds or even thousands of years ago, okay? My phone is going crazy. I did a postdoc at the Carnegie Institution in DC where I studied species interactions and dietary variation. And I realized that the tools I was using here, because my PhD advisor was a paleoecologist, could be applied to modern ecosystems, could be applied to study animals today and how they're impacted by human disturbances or how their diet and ecology changes over space and time, okay? And then I did another postdoc, and I realized that if I really wanted to do that, I should learn something about physiology. I should learn something about how animals process nutrients that they consume. And so my other postdoc was with Carlos Martinez del Rio and Dave Williams, actually, where I got really more involved in ecophysiology and took a look inside, underneath the hoods of animals to try to figure out what they were doing with all these things they were eating, okay? So the point of this is that I do this in marine and terrestrial ecosystems. I started out in here marine, and I was landlocked in DC, I was landlocked in Wyoming, and now that I'm in New Mexico, I'm still landlocked, so this poor Southern California surfer boy is still sitting in the desert. But never stop learning new tricks. Okay, this is the piece of advice I will give you. I can give you a lot of pieces of advice, but never stop learning new tricks. You might do a PhD on a particular subject and become an expert at that subject, but that doesn't mean that you can't do a postdoc on something completely different and learn a new skill set. And another postdoc on something completely different and learn a new skill set. And the first time you do a sabbatical as a faculty member, go somewhere and learn something new. So never stop learning new tricks. And isotopes are wonderful for this because you can figure out how to apply isotopes to anything. So if you learn that toolkit, look at Diane's career trajectory, Diane Pataki. She did not do urban ecology as a PhD student, not even as a postdoc, but now she's the urban ecologist, okay? So as long as you have this toolkit, you can do a lot of cool things. So what's that? Photosynthesis. I love it. The bear ecologist in the room from Slovenia says, oh, that's photosynthesis. Very good, Jeremy. I like it. Okay, photosynthesis. In photosynthesis is a lot of times, at least in terrestrial ecosystems, what's limiting? Water is limiting. Okay, we've talked about that earlier this week. All right. In marine ecosystems and aquatic ecosystems, carbon dioxide can be limiting too, but most plants on land, water is limiting. Well, what's that? Respiration. You're a plant ecologist, right? Oh, duh. So this is respiration, and normally what's limiting is food, right? And sometimes other stuff, oxygen, there's plenty of that in the atmosphere. So we need food. We also need some water as well for animals. But this is usually limiting. And I would argue that heterotrophs are really complicated in comparison to a lot of different plants because they use a lot of different types of sources of carbon, hydrogen, and oxygen for metabolism, energy, production basically, and growth to synthesize tissues. So not only this, which is carbohydrates, but they can use amino acids and they can use fatty acids as well. So your diet, as Turi was showing you today, any food product in the developed world, if you look on the back of that food product that you buy at the store, you'll see three things. You'll see the amount of protein in it, the amount of fats in it, and the amount of carbohydrates in it. Okay, you also see energy and salts and other things, but those different types of macromolecules can be utilized by animals in different ways to build tissues and to do work, to basically make energy to live. And that's what we're going to talk about today. So how do we study what animals eat? The first way we can do it is we can go out and directly observe animals. And this is awesome, but it only works for a couple different types of animals. So I studied marine mammals as a PhD student, and actually marine mammals are really difficult to study their diet. Why? They live underwater. And they swim really well. They're hard to follow. They swim incredible distances in short amounts of time, so us humans that are not adapted for a marine aquatic environment have a tough time keeping up with them, okay? There are a few marine mammals, for example, this one, sea otters, that I've done a lot of work on. This is my colleague Tim Tinker, that you can watch. Sea otters are unique because they go down, dive down, grab their food, always bring it back to the surface to consume it. So if you're sitting there with a high power scope and you know which otter you're looking at because it's been tagged with a telemetry tag, then you can compile some really awesome data on individual level foraging preferences. How many crabs, clams, urchins, snails, that particular otter, Emma here, actually that's the name of my graduate student, but she studies otters, so I just call this one Emma, how much she consumed in a given amount of time, okay? But this is a very rare opportunity. This usually doesn't happen, okay? And the cool things about this is that you can get really, like I said, very rich data, but it's really time and cost intensive. Tim makes a lot more money than I do. He spends a lot of time doing this. And in general, like I said, predation is difficult to observe. Another way we can do it is with scat or gut contents, okay? So we can follow animals around, we can pick their poo up, we can sift through their poo and figure out what they ate. Sounds fun. It's actually not too bad. It's better for some animals in comparison to others. Or we can wait till they die, we can rip them open and look at their gut contents, okay? So the cool thing about this is that you can usually identify different species of prey in both scats or in gut contents. So it gives you a lot of information about the diversity of diet in the animal. The cons are that it's snapshots. Your poo represents what you ate in the last few days, last day or two if you're regular, okay? And it's biased towards hard parts, right? Human poo, it's hard to do a scat analysis of human poo. We don't have a lot of hard parts in our poo. But a lot of animals that eat things that have bones and chitin and all sorts of stuff, that stuff will actually make it through the gastrointestinal tract and come out the other end, and you can identify that stuff sometimes to species, and that tells you something about what the animal ate, again, in the last couple days. This is one of my favorites. This is a killer whale that just pooed. This is a killer whale poo-sniffing dog on the front end of a boat. And when this guy poos, the dog gets excited. These biologists, poor souls, come by with a net and scoop up the poo. It's a pretty fun job. This is in Puget Sound. Okay? And then, of course, we can use isotope analysis. And this is really, I realized that we could, when I was a postdoc, my first postdoc, that this was really a tool, even though I had been applying it in the past, this is a tool that I can learn a lot about modern ecology with. And the pros are that it integrates over a variety of timescales, like Turi mentioned, depending on what kind of tissue you look at, whether you're looking at bone collagen, or tooth enamel, or breath, or liver, or muscle, or blood. All these different tissues in your body integrate ecological information over a variety of timescales, okay? The cons is that it's pretty low resolution. It does not capture prey taxonomic diversity, okay? Unless you're really, really lucky. So what you end up looking at, if you're an isotope ecologist studying animals is you look at prey functional diversity. You can tell isotopically a filter feeder in a marine organism from a grazer, okay? You can tell isotopically an animal that eats maybe kelp versus an animal that eats phytoplankton. But you don't necessarily, there's lots of animals in those food webs that do those kinds of things. You can't necessarily tease apart the animals that do that kind of specific kind of foraging. So keep that in mind. It's kind of like, I call it a dull spoon, but it's a very useful dull spoon, okay? All right, here's one of the first studies to do it. This is data from published in 1978, De Niro and Epstein. You've probably seen these names before in the last few days. Here we have a plot of animal carbon isotope values versus the diet carbon isotope values. That line there is the 1-to-1 line. So if you're on that line, that means you are what you eat. One thing I want you to notice is that most animals plot a little bit above that line, meaning that they're isotopically enriched in comparison to the foods they consumed, right? And their enrichment varies depending on what kind of animal you are and what kind of tissue you analyze. And that's what we're going to talk about today. So that's the general pattern. 1978, we knew this a long time ago. Here's another great carbon isotope-based study from 1978. This is probably the dawn of the use of isotopes in animal ecology. A guy named Brian Fry, who's an isopope, used to teach at this course. And he studied, as a student in Texas, he studied grasshoppers in West Texas. Really important and really diverse consumer in Great Plains ecosystems, grassland ecosystems. Again, we have animal carbon isotope value here on the Y, diet carbon isotope value here on the X, and he knew that you could take these numbers and then you could calculate the percentage using a mixing model, the percentage of C3 versus C4 diet items in a grasshopper's diet. And this is what he found. Pretty nice correlation, look at that. 0.97, 1-to-1, that's great. These guys, of course, you know now, I don't know why he plotted it this way, besides it was the 70s, and I don't know what Brian was doing in the 70s. These numbers are higher than those numbers. Today we'd know that you'd flip those around, right? As we discussed yesterday in class, negative 26 is a lower number than minus 14, so why switch the axes? But that's what he did it. But if you look at this carefully, these are the C4 loving grasshoppers. So these are the ones eating grass out there. These are the C3 loving eating grasshoppers, okay, so they love C3 plants. And then there's this one individual right in the middle, the most interesting individual in the study. It's a mixed feeder, okay? So it can consume both C3 and C4. And again, this is like, I think 20 or 30 different species here. Not huge sample sizes, because remember, to make these measurements in 1976 and 77, the year I was born actually, took a long time. So we don't have huge sample sizes, but there's a lot of interesting information in this graph.
How do you determine the diet C13 here? So you just assume they eat like one plant, or you just like assuming based on observation what they eat?
There's a lot of different ways you can do this. The main way, and actually I'm not showing this data, but Brian did it, was measure the plants. So you can go out there and measure the grasses, and there's a suite of different species, and you can put them all into a bin, C4 grasses, and that's the C4 isotopic composition. And then you can do all the C3 plants. So you have these two end members here, and then you can use a mixing model to partition basically how much of one versus the other they're consuming.
So then what's the value for the diet, delta 13C here? Is that from the animal?
No, that's something that he would directly analyze. So he analyzed the plants in the system. So he has data on the diet, the plants that these things are consuming, and then on the grasshoppers. I'm just not showing it to you. But trust me, he did. Here's another example of you are what you eat. This is one a little closer to home. This is my wife and my daughter. Okay? This is nitrogen isotopes.
Do you ever combine these data with like sequencing?
You're getting to the end of my talk. Yes. Hopefully we'll get there. I'm hoping. But yes. Yeah, we do. So metabarcoding to look at diet, or you can look at, we have a project right now on gut microbiomes. So we're actually identifying the gut microbiomes and looking at how they help animals deal with protein deficiency. But hopefully we'll get there at the end of the talk. Great question. So this is another example of you are what you kind of eat, right? Or you kind of are what you eat, and that is nitrogen isotopes. You guys have all heard the term you are what you eat plus 3 per mil or 4 per mil. right? It's a classic isotope term. Well, here's data for my wife in blue, nitrogen isotopes on the Y, days since birth of my daughter on the X. And yes, I am a smart guy. I made my wife give me her fingernails 80 days almost before Tessa was born. Right? So this is, you guys are like, this is like, what do you, I can't believe you're doing experiments on your own family, but yes, you have to do experiments on your own family. So this is what N is over time. This goes out to 440 days. I can extend this now out here because I'm still doing this experiment, probably will until Tessa goes off to college. And then the day, so it was actually really scary, I started taking fingernails shortly after Tessa was born. Try to take the fingernails off a baby that's this big. It's really a scary experience. But I did it. It's because I needed the data. I might have taken a little bit of blood too, but I needed the data, so I got my data. Look at Tessa, when she was born, looks exactly like mom, isotopically. Why is that? Yes, her food is coming through the placenta, basically, or not the placenta, the whatever the tube, the thing that I cut when she was born. I remember doing that, I just can't remember what it's called. Anyway, mom is sharing her food with her baby while the baby's inside her, right? But as soon as the baby's born, what happens in mammals? Mom starts nursing the baby, okay? And so almost immediately, although there's a little lag, and that goes back to what Turi was talking about this morning, but almost immediately, nitrogen isotope values increase. Okay? And then they sort of zero out right here at about 280 days. That's the apex of that curve. All right? And if you look at Tessa's values versus mom's value at 280 days, it's about a 2.5 per mil difference. So in this case, you are what you eat plus 2.5 per mil. All right? And you're like, wait, why is mom, what are you saying? Tessa's eating mom? But if you think about it, is this your question? Yes, breastfeeding. So yeah, it's nursing. Yeah. Actually, she nursed until about 365. She came back, my wife came back from a trip, a professional trip, a working trip, and she said, enough, no more. Baby is eating formula and solid food. We're not doing this anymore. So yes, but she was nursed for the first year of her life, almost to the day. And the reason why she's isotopically enriched in comparison to mom is that mom is using her own tissues, her protein and her fat stores in order to feed her baby. So she's catabolizing her own fat stores and her own protein. And any of you who have been mothers out there might have felt like this during nursing, is that your baby was eating you alive. And it's actually energetically more intensive to nurse a baby than it is to make a baby for a woman, for a female. So making the fetus, pregnancy, is not as energetically intensive as feeding that baby once it arrives in the world. Okay? You can also see that mom's isotope values are decreasing over time. I'm not going to get into that right now because I don't have time. It's an interesting observation, and it's pretty nice. It's only about 1 per mil, but it's a pretty nice decrease. Okay? So you are what you eat, plus 2 to 5 per mil.